molecule, and then we have also predicted the pharmacological activity of the small molecule, and we also generated the 3D structure of the small molecule. Now our ligand is ready. Now we have to um, prepare our drug target. So drug target, it can be retrieved from PDB. So how to know which is the drug target? So let's take for example, yesterday we were searching for Barangin from PubChem, and the predicted pharmacological activity for Barangin, it showed anti-neoplastic activity. So today we are going to find out the drug target that is responsible for anti-neoplastic activity, that can be used for anti-neoplastic activity, and then we are going to retrieve that particular drug target from PDB. So this is the URL for PDB, and the PDB's protein data bank. It has the 3D structure of all the biological molecules, including proteins, nucleic acids, and complexes also. Suppose if you have a receptor bound with a ligand, that complex 3D structure is also available in PDB, and it is freely downloadable, and we can download the protein and we can use the protein for our studies. And the next exercise that we'll be discussing will be predicting the binding site for the drug target. So that is very important, because we cannot bind our ligand to any part of any region in the protein. We have to bind, or we have to study the interaction of the ligand only exactly at the binding site of the protein only. So first we need to predict the binding site. So binding site can be, it is also available in the UniProt database, that is a protein sequence database. In some cases, the protein binding site information will not be available in UniProt. So in that case, we need to predict with the server called as CAST-P. So this is a computed atlas of surface topography of proteins. How this particular server is predicting the binding site is, it predicts the pockets, and then it tries to identify what are all the regions or which amino acid is exactly responsible for binding. So here we have to input the PDB ID, that is protein data bank ID. There is an identification number that is associated with any protein entry in PDB. So we have to mention the PDB ID. Depending upon the PDB ID, this CASP server will be predicting the binding site region. So after predicting the binding site region, today next exercise that we will be discussing will be performing our molecular docking. So actually, whatever experiments that we have seen till now will be a very simple experiments that we need to copy and paste somewhere, and you will get the result by click of a mouse. That is very simple. But when you are doing performing docking, it requires a series of steps. Each step need to be followed carefully, and only then we will get the correct docking. Otherwise, each and every step we might end up with an error. It has a series of steps that has to be performed, and we have to follow each step carefully. And there is also some command line operations that we need to do. So it is very simple only. So I request each one of you to kindly carefully notice what is being demonstrated, so that it will be useful for you when you do it independently after the workshop. So so this Autodoc tools, it is nothing but it is actually a suite of tools. So suite is nothing but you have a collection of programs. It is not a single program. We have a collection of programs for visualization, for predicting the binding site, so for binding affinity, and to know about, to analyze or visualize the complex. So it has a suite of different tools. It is a suite of many different programs together. And the main two programs that is available is in Autodoc 4 is Autodoc and AutoGrid. So what is the purpose of Autodoc? It predicts the docking of the ligand to the excuse me, to the target protein, and AutoGrid, it calculates these grids. So grids are nothing but they are imaginary lines that is being drawn across the binding site, so that the ligand will be able to exactly pinpoint the binding position, and then it will be able to interact with the drug target. So that is a imaginary grid. It is being developed by AutoGrid, and with the help of the grid only, the ligand will be able to interact with the drug target. So these two tools are very important. And what are all the requirements for performing a molecular docking calculation using Autodoc? First, we should have a 3D structure of the receptor. So in docking, we call the macromolecule as receptor, and we call the small molecule as ligand. So here you don't mean that receptor is some receptor protein like GPCR, but here in docking we call the macromolecule as the receptor and the small molecule as the ligand. So we will be retrieving the 3D structure of the macromolecule from PDB, and the 3D structure of ligand yesterday itself we have generated, either searching from databases or by sketching the molecule and generating a 3D structure. And you should also have an input for the binding set. So you can directly search for the binding set in UniProt database, or you can do a prediction of the binding set with the help of the CAST-P server. So these are the requirements for performing a molecular docking calculations. So you might also ask a question, suppose if you don't know the binding site and if it is a modeled protein, so what can be done? So if you don't know the binding site, then we have to go for a type of docking called as blind docking, where we give the freedom of finding the binding site to the AutoDock tool itself, the docking software itself. So docking software itself identifies the cavities and the pockets inside a protein, and it tries to find out the complementarity in shape, charge, and all the properties for the given ligand, and it tries to fit the ligand to the drug target. So that is called as blind docking. So blind docking can be done only if the information about the binding site is not known. But if the binding site information is known, then it is always a good practice to specify the binding site in the docking software so that the binding can be biologically correct. So that is the important point that we have to keep in mind while doing a docking, molecular docking. So these are a few steps I just wanted to mention here. So these steps we'll be quickly doing in demonstration. So there are four important steps we do with while performing a molecular docking with AutoDock tools. First one is we need to prepare the receptor. Second step is we need to prepare the ligand. Third step, we need to prepare the autogrid file, and then we should also prepare a AutoDock file, also parameter file. So how to prepare the receptor? The first step is the receptor. Suppose if your structure has been crystallized by NMR technique, if the structure has been solved by NMR means, then sometimes water molecules will be present along with the PDB coordinates. So the first step, we need to remove the water molecules, if any, in the macromolecule, and then we need to add hydrogens, and then we have to save the PDB file. So that will be the step involved in preparation of the receptor. So we have to prepare the receptor by following two important steps. One is removing the water molecule that is being available in the PDB structure. The second step is adding hydrogens. So why we need to add hydrogens is in PDB, hydrogens will not be explicitly represented. So hydrogens will be generally hidden. So only if the hydrogens are present, the hydrogen bonds can be detected by the docking software. So that is why we are adding hydrogens to the PDB file. So after following these two steps, we need to save the file in PDB format. The second step is a preparation of ligand. So we need to prepare the ligand in that is a small molecule. So here we have a separate menu for ligand, and we have to open the ligand and we have to save the ligand in a particular format called as PDB-QT format. So PDB-QT format is nothing but the charges to the atoms will be added. The partial charges that is available in the atom of the small molecule will be added, and that format we call it as PDB-QT format. So the charges, the partial charges available with the atoms in the small molecule will be added, and we have to save the file in a format called as PDB-QT format. Next one, we need to create the autogrid parameter file. So for creating the autogrid parameter file, first we have to choose the macromolecule. So the autogrid program needs to specify the macromolecule, and then we also need to specify the ligand molecule, and then we have to set the grid for center binding site. So where is the binding site? We have to center the grid for that particular binding site, and then we have to save the file using an extension called as GPF. So .gpf extension we need to save. So GPF is grid parameter file. So we have it is called as grid parameter file. So to create this file, first we need to choose the macromolecule for which we are going to perform docking, and then next we have to choose the ligand molecule. Then we have to set the grid for the binding site, and then we need to save the file using GPF extension, that is .gpf is grid parameter file, and then we have to run the autogrid program. And final step is we have to create the AutoDock parameter file. For creating the AutoDock parameter file, we need to choose the macromolecule, then choose the ligand, and then we need to select the parameters. sorry for the spelling mistake, it is not paraments. We have to select the parameters, and then we have to save the file using .dpf extension. So dpf is docking parameter file. So with the help of autodock menu, we will be creating a docking parameter file, and then we have to save it, and then we have to run the autodock program. So these are the steps that we will be following. I will slowly move through while doing the demonstration, but just note that these are the 4 important steps while doing. So how to run the AutoGrid and Autodock programs. So this also I will tell you. So we have to go to the command line. I will just show you how to open the command line, how to execute these commands. So this is the command that we have to specify. So Autodock tools, it also has menu-driven running of the program, but many of the CPUs does not support running it with the help of a click of the mouse. So it is little easy, or it is actually very good to do it in the command line, because many of the CPUs does not support. After creating all the things, when you try to run the AutoGrid program, it may give it some error. So it is always good to run your AutoGrid and Autodock programs in the command line itself. So the command will be AutoGrid 4, and there should be a space and minus p. Minus p means we are telling that it is a parameter file, and then we have to give the grid parameter file. So we will be saving the grid parameter file, and we have to give the name of the grid parameter file, and then minus L. Minus L is the log file. So that will be the output file that will be saved. So that should be saved in an extension called GLG. So that is grid log file. So we have to name it as we have to give some name, and then save it using the extension .glg. And then after issuing that command, it takes a few seconds to run the AutoGrid program. After that, we need to issue the Autodock command. So Autodock command is similar to that, but instead of AutoGrid 4, we have to give Autodock 4, and then minus p is the parameter file, and then you have to give the name of the Autodock parameter file that we have created using Autodock tools. And then we need to give minus L, that is a log file, and then we have to give the name for the docking log file, that is .dlg extension. So this is how we run, and then we will be getting the result, which we will be analyzing tomorrow. So now I will start with the demonstration. So the first step, as discussed, I told we need to identify the drug target. So the very important method, or the most popular method of identifying the drug target for a particular pharmacological activity, is to search in the literature. So we can search in the literature and identify which protein is actually involved in that particular pharmacological activity. We can study the mechanism of action, and then we can choose the drug target. But there is also one simplest way by which we can identify the drug target. So that is searching for drug target from a database called as drug bank. Okay. So just go to Google and then type drug bank. So why we are going to drug bank is a database of the FDA approved drugs. Actually, yesterday last exercise should have been this one. So since I wanted to work with all the drug targets today, I did not touch drug bank yesterday. So this is a database which has all the information about all the details about drugs that is approved. So here also you will be having a search box. We can search for any drug. So you can give the name of any drug and see what is the information that is available. So rifampicin, if you try, it will give you the summary of that particular drug. So what are its brand names? What is its generic name? So what is the background information? And then you can also download the structure. So what is its weight? All information, pharmacology, what is the pharmacology? So how it is being, what is the toxicity? What are the adverse effects? So like that, we can get all information about any particular drug that we are searching. So drug bank is a compendium of all the available drugs that is there, and including the structures. You will also have all possible activities, that is mechanism of action, how it is targeting, whether by inhibition or modulation. So what are its what dosages it can be given. So all those information, everything available about the drug will be available in the drug bank. So now we are going to use this drug bank to identify our drug target. So So what we can do is, instead of searching for the drug name, we can search for the pharmacological activity. So why I am using drug bank? Because I am going to find out the drug target for antineoplastic. Yesterday we have created one. We have downloaded Barangin. So it is from PubChem. So we have predicted its pharmacological activity. It showed antineoplastic, that is anti-cancer activity. So today we are going to find out the drug target for antineoplastic activity. So for that, what we are doing it in an indirect way. How we are doing it? We are searching for the drugs available for antineoplastic activity, and from the drug name, we are going to search the drug target. So that is what we are going to do now. So if you see, you will have a list of 725 different drugs are available with anti-neoplastic activity. So let us read the information. So for let's take for example otiracil. This is an adjunct to anti-neoplastic therapy. So it is actually not it is not having anti-neoplastic activity, but it is used as an adjunct. So we cannot use this particular drug. But if you take the next drug, that is testolactone. So it is having an antineoplastic. It is an antineoplastic agent, and it is having it is a derivative of progesterone. So let me search for this particular drug, and from that, let me search for the drug target. So this is why we are following this method is, if we are searching for literature, then we need to actually know the actual mechanism of action. Then we have to use the drug target, and then we have to search for the drug target in the PDB database. If the structure is not being solved, then we cannot proceed with that particular drug target. But this one, anyway, we are going to test our pharmacological activity with a standard drug. So we also need a standard drug. So we are taking the standard drug and its corresponding drug target also we are choosing from the drug bank itself. So that can be utilized for testing our compound under study. So which compound we are going to prove it is having anti-cancer activity means, for that compound we need to choose a drug target. So now we have got a drug called testolactone. So this is a medication used to treat advanced breast cancer. It is given that, and you also have you can also download the file also. So let me download it. So here you have different formats. Yesterday itself I told you, for doing molecular docking, we require the structures in a format called PDB. So let's download that. So it will not be downloading as a separate file. It will be coming as in the browser itself. So what we need to do is, we need to copy that particular file, and then we need to save it in a notepad. So let us save it in notepad. Let us save it in notepad, and then let us save it. Go to file, give save. So yesterday I have created one folder called docking in desktop. So inside that itself, I'm just going to save it as drug. And then there's an important point that we need to follow. So we know that in Notepad, all the files will be saved using .txt extension, but we need the file in PDB extension. So what we should do is, we need to select all types. So we have to save the type as all files, and then in the drug we have to give drug.pdb. So we are manually giving the extension for this particular file. For that, we need to select all files. If you don't select all files, the file will be saved as drug.pdb.txt. So if you give like that, the Autodoc tools will not be able to recognize our file. So for Autodoc tools to recognize our file, the extension should be only .pdb. So how to save that? Just we need to go to save as file type. We have to mention all files, and then we have to give the name of the drug.pdb, and then we need to save. So now what all is ready with us? We have the ligand, we also have the standard drug also, but we don't have the drug target. So only if we have the drug target we can go for molecular docking. So how to search the drug target? So we have antineoplastic agent. So we have a testolactone. So if you see the menus that is available in the left hand side, so that will be a menu called target. So this is an approved drug. This is available in the market, and they will also mention what is the target which this particular drug is going to bind with. So this is a cytochrome P450 19A1 protein, and the mechanism of action is inhibition. So this drug is generally inhibiting this particular protein, and it is also giving the specific function also. So this information is there. So we can search for this particular protein, and then we can get the PDB. So if you see here itself they give the UniProt ID for that particular protein. So UniProt is a protein sequence database. So UniProt is actually a protein sequence database, and actually we require the 3D structure of this particular protein, the cytochrome P450 19A1. So we can directly go to PDB and we can search for this protein, but we will get multiple entries sometimes, and it will not we will not be able to fetch the right protein. So it is always good practice to search for that particular protein from the sequence database, and from there to identify the PDB. So here we have the UniProt ID. So that is P11511. So let's go to UniProt database. So UniProt is a protein sequence database most of you might be knowing. So this will have information about the protein sequence information. So what is the UniProt ID? UniProt ID is P11511. So P11511. So let us enter the name of the protein. So it is all, these are the things that I am doing so that it will be easy for you to practice later on, because if you try to search for the protein directly in PDB, then you will not be able to fetch the right protein, because it is showing cytochrome P450 19A1, but its other name is aromatase. It is an aromatase enzyme. So see here it is having cytochrome P450 monoxygenase activity. So there will be many synonyms for one particular protein, and each database will be maintaining a separate name. So if you search using cytochrome P450A11 means 19A1 means the PDB will not show you the exact protein that you will be requiring. So that is why I request each one of you to note down the UniProt ID and go to UniProt, and from there you fetch the protein structure. So that is the simplest way. So if you see, this is the UniProt database. This is a protein sequence database. It also gives you the catalytic activity. It gives you what is a cofactor, what are all the enzyme kinetics. See, this is a very important feature. It is showing the substrate binding site also. So if you see 309, the residue 309 is actually the substrate binding site of this particular protein. So we can note down the binding site from here itself, from UniProt itself. For some UniProt entries, the binding site will not be available. In that case only, we have to go for the next one, that is CAST-P. That also I will demonstrate at the end. But for now let's see that we have, let's note down that the binding site of this particular protein for this enzyme is 309. That is the region. This is the amino acid number where the substrate is actually binding. So now when we want to target our ligand or small molecule, then we should select the binding site around the 309 region only. Okay. Now many information will be available. So name, taxonomy, pathology, natural variant, all information related to the protein sequence will be there. If we come at the end, we have the structural information. So you have a list of structures that has been available in PDB, but choosing the structure is very important. So first let us see the first one. The PDB ID is 1TQA, but see the method of solving is model. So it is a computationally predicted model. We will be using that only if the experimentally solved structures are not available. If experimentally solved structures are not available, then we can use the model. No harm in that. But for this particular protein, experimentally solved structures are available. Many experimentally solved structures are available. So if you see the first one, 3EQM. So this is the PDB ID for this particular protein. That is 3EQM. Let us note that also. So, and then we have the X-ray method and the resolution, what is the chain and what is the positions that is given, that is also there. So now we can have it. So we can directly download it from here. So now UniProt is having an option to download the PDB structures directly from UniProt itself. So now we can directly download. Otherwise, we can go to PDB and then we enter this particular PDB ID, and then also we can download. Now I'm going to download from UniProt database itself. So where should we save it? So we have a folder called docking in desktop. So open that. Then let's name it as, this is our receptor, right? So let us name it as receptor. So I will quickly give a recap of what we have done till now. So we have gone to drug bank to identify the drugs with antineoplastic activity. And from that drug information, we have identified the drug target. And then with the help of the UniProt ID that is available in drug bank, we have downloaded the PDB format from UniProt database. That's what we have done till now. So if it is clear, we can move on to the next. So it is saving in .ent extension. So if you want it in PDB extension, we can go to PDB. So this is the RCSB PDB is the PDB homepage. So what is the PDB ID? We have noted down. It is 3EQM is the PDB ID. So search for that. So see, if you see crystal structure of human placental aromatase cytochrome P450 complex with androstenedione. So this is actually a complex with one protein and one already ligand is already bound to that. This is a complex. Let's download that. So we require it in PDB format. So let's go to PDB format. So okay. So now you see it is saved in PDB format. So that is the required format. So when you download it from UniProt, it is saving in .ent, but that is not supported in Autodoc. So I have come to PDB and then I have downloaded the protein in PDB format itself. Okay. So we have the drug target now. So before opening Autodoc, we need to do one thing. So if you see this particular information, so this particular protein is actually a complex which is having one small molecule androstenedione, which is already available here. So now what is the thing is we need to remove the natural ligand from this particular complex because then only our binding site will be free for our drug to bind to it. So the first thing is we need to remove the small molecule already attached in the PDB entry, and then after removing only we can present it for docking. Otherwise the binding site will not be empty and the ligand will not be able to bind to the protein. So how to do it manually? We have to do it manually. So let's go to desktop. So let's open the PDB file. So let me delete the TNT file, otherwise it will be confusing to us. So this is our PDB file. So please right click on it. So give open with WordPad. So because we are not opening the structure, we are not going to visualize the structure. We are just going to delete the small molecule which is already attached with that particular complex, because then only our binding site will be free for the ligand to bind to it. So if you see, this is how a PDB file will look like. So all the experimental information will be available, when it was crystallized, what are its other names, what is its enzyme classification number, which organism it has been taken, what is the taxonomic idea, from which organ it was taken. All those information will be available for this. So these are all the crystallographic information, how the crystals were generated, crystallographic information will be symmetry, all those information will be available. Then coming down, we will be having the sequence information also. So this is the sequence information, so methionine, valine, leucine. So all the sequence information will be there. And then it also shows the secondary structures also. So where the helix is formed, so how the sheet is, where the sheet is formed, all the secondary section information is also available. And coming down, so this is how the atom will be stored. So whenever you want to visualize the 3D structure, means we should require the coordinate information. That is where each and every atom is present in the XYZ coordinate. So you should have a X coordinate, you should have a Y coordinate, and you should have an Z coordinate. Then only the 3D information will be available. So here, if you take the first atom, atom number 1 is a nitrogen that is belonging to serine of chain A, 45th residue. So serine is a 45th residue, and then the first atom, it is having the X coordinate, Y coordinate, and Z coordinate. Similarly, for our next atom, so Ca is C alpha atom of serine again of 45th position serine, and you have the X coordinate, Y coordinate, and Z coordinate. So like that, we will be having the information for all the atoms that is present in this particular protein. So only if this information is available, we can visualize the 3D structure of that particular protein. So this is the work of X-ray crystallography. It identifies the positions of each and every atom, and then it displays, or it produces the 3D structure of the macromolecule. So why we have opened this file now? We just wanted to remove the small molecule which is naturally attached to it. So to remove that only, we have opened this particular PDB file. So for that, let's come down. So it will be at the end. Okay, we come down, if you see. So we have the atom till here. So this ter represents the termination. So termination, so 3,659 atoms are there. The last one is actually asparagine. ASN is asparagine 496. So the second, from the after the term, we have a term called het atom. HET-ATM. HET-ATM is heteroatom. So this is the one which we need to remove so that our binding site will be free for the ligand to move. So we will select all the HET-ATM. Please do not come till connect. Just select only the HET-ATM entries. So wherever there is HET-ATM, so you just select it and then just press delete. Just we need to delete. Please don't delete any of the connect information. If you delete the connect information, then the protein will not be available. Then we will save it. So what did I do? I just downloaded a PDB file from PDB database. I opened it using WordPad, and then I searched for heteroatom. So heteroatom entry will be given using HET-ATM, and then we need to select all the HET-ATM entries, and then we need to delete all the HET-ATM entries without disturbing the connect information. So after that, we will save the file. Now we have the ligand, we have the ligand 3D structure, we have the PDB 3D structure. So now let's start docking. So hope it is clear till now. If there are any clarifications, you can give in chat box. Otherwise, we will move on to docking. Yes. Any doubts? Shall we move on to docking? Okay.
Yes, ma'am.
Yeah. Thank you so much, ma'am. We'll proceed. So we have our protein as well as we have the 3D structure of the ligand ready. We'll go for docking. So how to download the Autodock file? So we are going to do docking with Autodock. So Autodock, there is a tool called MGL tools. So MGL tools, these tools will help us to visualize because Autodock is a command line software. So to make it user friendly, they have developed a tool called as MGL tools. So we can go to that and we can download it. So Windows version, we can download it and we can install it. MGL tools. The first thing is we need to download and install the MGL tools, and then next we have to download and install Autodock also. So MGL tools are supplementary to Autodock. So this will help us to do most of the activities using click of the mouse itself. So MGL tools will help. It will be a GUI interface, that is graphical user interface for the Autodock program. So we will be downloading this MGL tools and then we will install it. This will be a supplementary to our Autodoc program. So we have to download it and install, and then we should also download the Autodoc also. So just go to Autodoc and download Autodoc 4. So both of them are free, and whatever activities that we are doing will be can be published also. Whatever docking we are doing with Autodoc is acceptable for publication also. So so whichever version of operating system you are having, depending upon that, you can download the software. So we can directly download and we can install, and then you will after installing you will have the file like this. This will be the Autodoc window. So you will have a couple of menus. There will be two menus, one separately for macromolecule and one separately for ligand and performing the docking. So this will be, if you have installed MGL tools and Autodoc, after installing you will have like this. Suppose if you don't install Autodoc, this particular menu will not be available. That is ligand flexibility. This menu will not be available. So from that you can understand that you have not correctly installed Autodoc. So you have to install both MGL tools as well as Autodoc. Only then you will get both the menus. Only then you will be able to perform docking. Okay. So now let us before starting with the docking, this is a simple hack I just wanted to teach you. Because since we are going to perform, we are going to run Autodoc and AutoGrid programs in command line. Let us, to make our path easier, let us take the Autodoc files and place it in the working directory. Okay. So after installing MGL tools and after installing your Autodoc, your C colon will have a folder called program files. So program files will be already there in your C colon. After installing Autodoc, you will be having, okay, there is a program files x86. Please go to that folder. Inside that folder, there will be a folder called as the Scripps Research Institute. Okay. So there will be a folder called Autodoc. Then there will be another folder called 4.2.6. There will be two executable files. Let us copy those files and place it in our working directory. So this is optional only for those who can give the commands, those who can set the path in the command line itself. They need not do this one, because being biologists, we are all afraid of computers sometimes, and that is why I just wanted to teach you this hack. So let us copy the executable files and then let us paste it in our working directory. So that is our docking directory. Let me paste it. Okay. Okay. Since we are having drug separately, we are having ligand separately. So let us create a folder for ligand separately and then for drug separately. So these are all to avoid confusion, because if you are performing docking, the output, all the output files will be with the same name. So to avoid confusions and to avoid errors, we will be using these small hacks. So inside that ligand folder, I'm just going to copy ligand, receptor, and then AutoGrid 4 and Autodoc 4 files, and I'm going to place it in that particular folder. I will repeat this step once again. So after installing MGL tools and Autodoc tools, inside our C colon, we'll have program files and as well as program files x86. In some operating systems, the Scripps Research Institute will be available in program files itself. Please search there. Otherwise, it will be saved in program files x86. Inside that, you will have a folder called the Scripps Research Institute. Inside that, we'll have a folder called Autodoc. Inside that, we'll have another folder 4.2.6. Inside that, we'll be having two executable files. Please do not do anything with that. Just copy those files and paste it in your working directory. So here we have the ligand, and inside that we have the macromolecule. This is the receptor, and this is our small molecule. And then we have the AutoGrid file and Autodoc file. So all the four files are copied here. Now let us run our Autodoc program. Let's go here. So what is the first step? I think all of you might be remembering. We need to prepare the macromolecule. We need to prepare the receptor. So how to prepare the receptor? I told you there are two steps. One is first we need to remove water molecules if there are any, and we need to add hydrogens. These are the two steps that we have to do while preparing the macromolecule. So how to do that? Just go to file, just give read molecule. So where is our macromolecule? So we have desktop, we have a docking folder. Inside that, we have a folder called ligand inside that. So we have to be careful enough. Here we have named our macromolecule as receptor and the small molecule as ligand. So let us select receptor. So just give open. Okay. So this is the structure of that particular protein, that particular macromolecule that we have. So we can also have displayed in ribbon model. There is an option to show the ribbon model. So, so different options are there here. So let that all can be done, or it may not be done also. So simply to show you, I'm just showing. So we have loaded the macromolecule. Now we need to delete water. So for that we need to go to edit menu. So there is an option called delete water. So if there are water, we will be having some yellow and red color atoms that is be displayed here, that will be present around the surface of the protein. If it has been NMR, it has been solved by NMR. But our structure is X-ray crystallographic structure only. It is not having any water molecule, then there is no need to delete. But still, how to delete if water is present means just go to edit menu and click delete water. So if there are any water molecules that is present, delete. So why we need to delete water molecules means because that it will interfere with the binding of the ligand with the um, receptor. So to avoid that interference only, we are removing the water molecule. So just go to edit and delete water molecule. The next step is, I told you, we need to add hydrogens. So why are we adding hydrogens? We are adding hydrogens only then hydrogen bonds will be formed. So how to add hydrogens? Edit menu, hydrogens, add. So we can add all hydrogens or we can add only polar hydrogens. But sometimes we will be requiring all hydrogens, or so it is always good to select all hydrogens. So click all hydrogens and click OK. So see, so some white color small dot like things have come here. So those are all hydrogen atoms. So we have prepared our receptor now. Just we loaded the receptor, we deleted the water molecule using edit menu, and then we added hydrogens using the edit menu. Now we need to save this particular receptor in PDB format. So just go to file. There is an option called save. So there we need to select write PDB. So click write PDB. So here we need to save all the things in the working directory. So in which directory we are going to work, there only we need to save all the things. So just give browse and just go to desktop. So go to docking, ligand, and instead of naming different files, let us name it as receptor only. So select receptor and give save. So it will tell, do you want to replace it? Yes, we need to replace because we have the same receptor, but we have added hydrogens. That's it. So let us name it as a receptor only. Click yes, and then click OK. And then give yes. That's all. It is asking us whether it can be overwritten or not. So what did I do? Just file, read molecule, then go to edit, delete water, then hydrogens, add. And then what did I do? In file, we have save option, save, write PDB, and I have saved that file with the same name as receptor itself. Now our receptor is ready. What is the second step? Second step is we need to prepare our ligand. We need to prepare our small molecule. So how to prepare it? So here there is a separate menu at the down. We have to, for loading our ligand and preparing our ligand, we have to go to ligand, input, and give open. So where have we kept our ligand? It is in our working directory. Go to desktop, docking, ligand. So it is not showing any ligand here because it is asking for PDBQT files. If you see here, the file type is displayed as PDBQT, but we have our ligand in PDB format. So let's click on it and then let us select PDB files. So now our ligand and receptor will be displayed. So which is our small molecule? So ligand is our small molecule. So let us select that, then open. So see here, so our small molecule has been displayed here. So this is our macromolecule, this is our receptor, and this is our small molecule. So we have we have loaded our ligand from the ligand menu. So from ligand menu, click input and open, and then choose the file type as PDB, and then export the file to the working place. Now we need to save that ligand in PDBQT format. I told you PDBQT format is adding charges to the atoms. So if the atoms have partial charges, it will be added and that particular format will be saved. So save as PDBQT. Go to desktop. Okay. Save. So already PDBQT is there. Just save it as PDBQT. So just that's all. So here we just added the charges and then it is saved in the PDBQT format. So the first two steps is over. The third step is preparation of the grid parameter file. So for the grid parameter file, we need to follow a series of steps. First, we need to choose the macromolecule. Then we need to choose the small molecule. Then we need to prepare the grid box. And then we need to prepare the, we need to save the grid parameter file. So let us go to macromolecule and then give choose. So this is a very important step. Sometimes people will click open, but our macromolecule is already opened. So we need to just choose the file. So from macromolecule submenu, just click choose. So a window will be open. So from there, we need to select receptor, just click receptor, select molecule. So now the macromolecule has been selected by the grid. So it will ask you to add charges. So after adding charges, it will ask us to save in PDBQT format. So where should we save? Just go to desktop, go to docking, we can then have receptor.pdbqt. So after doing that, you will have the file becomes, the macromolecule becomes gray color. So from that itself, you can understand that whatever steps we have done till now is correct. We have correctly followed all the steps. Only then this gray color will come. Otherwise, it will not appear gray. It will be appearing in the multicolors only. Now we have chosen the macromolecule. Next, we need to choose the ligand molecule. So for that, we have to go to set map types. Under that, we need to choose ligand. So choose ligand. So again, the small window will appear. So from there, let us select ligand. So select ligand. So ligand will be selected. Now, the third step is we need to generate a grid box. So please, all of you, please see the screen. There will be a grid box that will be appearing. I think all of you can visualize the grid box. So there will be a red, blue and green color box. Three-dimensional cube will be appearing around the binding site. So at this here only we need to center our binding site. So this will be automatically, Autodoc will automatically identify the pocket and it will place the grid box in the cavity and pocket. But still, if you want to exactly center your grid box around the binding site means we need to change the dimensions. So I will tell you how to identify the dimensions. So we have downloaded our macromolecule PDB file here. And we have already noted down the binding residue number. So I think the binding residue number was 309. Okay. So let us first find out the residue 309. So this is the atom number. The first one is the atom number and this is the residue number. This is the amino acid number. So our amino acid number will be 309. We need to take that. Okay. And then we need to find out the XYZ coordinates. So 309, see, so this is the coordinates are present from here 309. So 309 is an aspartic acid, it is ASP, and it is having many atoms 1, 2, 3, 4, 5, 6, 7, 8, 8, excepting hydrogen. Totally 8 heavy atoms, 8 atoms are present. So let us take one coordinate, let's take for 82.833. We need to note it down. So we are going to center our binding. So 82.833, that is X coordinate, and Y coordinate is 48.139. 51.523. So out of these different atoms, you can choose any of the one that you can take. So because it will be oriented nearby only. So here I have noted down 82.833 as x coordinate, 48.139 as y coordinate, and 51.523 in the z coordinate. Now we need to input this in our docking grid file. So this is our grid file. So we need to change it here. See, I already told you, depending upon the cavity, it will automatically orient the x and y. So x, y, z are almost, it is nearer to the value that we have already noted. But still, let us exactly change it to. So what is the x coordinate that we have noted? X coordinate is 82.833. So you can see the change in the position of the grid box as it change the values. So now let me change it to 48.139, that is the y-axis, and the z-axis will be 51.523. So now this is how we mentioned the binding site orientation. So now our grid box will be positioned that center to that of the binding residue. So this is it will be centered. So what is the point of giving the grid box means? It is an imaginary box. Around that only the docking will be taking place. The software will be able to identify the binding residues only at that particular region. So we are providing an imaginary space, 3D space for the ligand to occupy itself to identify the complementary properties, atoms with complementary properties, and then it will bind to itself, to the macromolecule. So that is how we center. Now we have, after specifying the grid box parameters, then we need to save the grid box parameter file. Just go to File, Close Saving Current. So once I click this, the grid box will become invisible. So please, click Close Saving Current. So that's all. So now we have set it. So I will once again repeat the grid options. So first we need to go to macromolecule and then we choose the macromolecules. Once we choose the macromolecule will be saved in PDB-QT format. And then to choose the ligand, then we need to set the map type and then we have to choose the ligand. And then we have to go to grid box and then we have to center the grid box around the binding site residue, and then we can. Suppose if you have a question, I don't know the binding residue, my protein's cavity is not identified, I have a model. In that case, what we can do is we can just do a blind docking. So blind docking, how you can do, if you change this particular position, your grid box will automatically become bigger. So maximum is 126. Again here, if you improve it, it is 126. Here also in x-axis if you do. So it will cover the entire protein. In this case, you need not mention the binding site. It will do a blind docking and it will try to identify where the binding residue is there. But since in our case, we actually know the binding site, let us not do, because it takes much time. Let us not do that. Let's go to we have the binding site information. So let us make it as 40. Just by dragging, we can change the parameters. Okay. Now let me save the file saving current. After doing this, we need to save the grid parameter file. How to save the grid parameter file? Go to grid, click output, then save GPF. So where to save? Go to desktop, go to docking, then go to ligand. Let me name it as complex. So this is what I'm going to do. So this is a complex and the .gpf extension will be given by the Autodoc tool itself. So GPF, I told you, grid parameter file, it is saved. So we have completed grid parameter file generation. Next, we need to create the Autodoc parameter file. So for creating Autodoc parameter, just go to docking menu, go to macromolecule, set rigid file name. So here it will open a window and it will ask you to select the macromolecule. So go to desktop, docking, ligand. So which is our macromolecule? So we'll be having the receptor in PDB-QT format. Select the PDB-QT format. And then go to ligand and choose. So once you click choose, you'll have ligand. Select ligand. So it will give you parameters, what are all the different parameters it is. So what is the center that of the ligand will be present, how many torsions are present, so how to do the rotations, all those information will be there. Give accept. Go to docking. Now we need to search the search parameters. So search parameters, there are three important algorithms. One is genetic algorithm, simulated anemia, and local search parameters. We will be choosing the genetic algorithm. It is an efficient algorithm and it runs the docking procedure very quickly. So just give genetic algorithm and then give accept. So these are all the different parameters. So what are the different runs? What is the population size? What are the number of evaluations that will be done? Number of generation, rate of crossover. So this genetic algorithm is basically based upon the biological genetic concepts only. That is why it is called as genetic engineering. Crossover, mutation rate, all those information will be there. So this is nothing but different permutations and combinations of identifying the different orientation in which the ligand can bind to the drug target. So I told you, so ligand will change its position so that it can fit exactly to the binding site of the macromolecule. So these are the different ways by which the orientations will be generated. So for that only genetic algorithm is used. So these parameters are given and then give accept. Then finally, go to docking parameters. We will be having what is the random number generator, what are the energy parameters, step size parameters, all those things we will be using the default for now. If you want to do advanced, we can do later. And then finally, we will save the docking parameter file. So here we will be choosing Lamarckian algorithm. Here also you have genetic algorithm for doing, or we can also do simulated aligning also. It is doing Lamarckian genetic algorithm. This is a type of genetic algorithm only. So this we will choose and then we'll save it. So this also I'm going to name it as complex. So the grid parameter file was saved as complex.gpf and docking parameter file is saved using complex.dpf. dpf is docking parameter file. So it is saved as docking parameter file. Sorry. So we need to save it as complex.dpf. We need to name it as complex.dpf. Save. Grid parameter also we need to save using the extension. So, GPF. Not accepting. So we will mention the extensions also. Now we have completely prepared our parameter files. We need to run the programs. So there is an option here to run autogrid and run autodoc. So this system does not support running it in the user interface. So I just need to run the command. Suppose if your computer is supporting, then you can directly click run autogrid and you can directly click autodoc after running the autogrid. But I need to, most of the machines does not support and it will, our systems will not be of that capacity and then it will start to hang. So I'm just going to run the command in command line. So it's a very simple one. Just go to run and type CMD. So a command prompt window will be appearing. We need to click on that. So here it will come with C users chunk. So this is the, so what is our working directory? So we need to go and find out. So this is our working directory. Our working directory is so desktop working directory. So let's right click on our working directory and see the properties. So properties is C. So this is the complete address, C user chunk, OneDrive, desktop, docking. So this is the path of this particular file. So we need to go to this particular folder and then only we can run our Autodoc. So let's go to the command line. So now our working directory, our command prompt is showing the folder user C uses chunk, but we need to go inside OneDrive. So how to do that? So there is a command called cd. Cd means change directory OneDrive. So change cd space OneDrive. So now we will have the address C users chunk OneDrive. Now it has come to this particular path. Next we need to give desktop. So let's mention cd desktop. So now see the path has come here. So see user chunk OneDrive desktop. Now we need to go to document. So cd document. So where is our all our files are there, or all our files are inside Ligand directory. So cd Ligand. So this cd is the same command. That is, we need to change directory. We need to go inside one directory. Means we have to give cd space and the name of that particular directory. Now we have come to our working directory. Now we need to issue the command. So how to issue the command? Autogrid 4. This is the command. Autogrid 4 space. We need to give -p. So what is the name of the grid parameter file that we gave? Complex dot gpf. Okay. Space -l space complex dot glg. So this is the command that we need to issue. So autogrid 4 space -p stands for parameter, and we need to give the grid parameter file name of the grid parameter file complex.gpf space -l complex.glg. So glg is the log file, grid log file. So this output file will be created in this name complex.glg. So after issuing this command, we need to enter. It takes little time for running this particular command. After that only we need to issue the autodoc command. So okay. If there are any warnings, then that will be given. Okay. So now after this, or after running this command, then we will have to run another command autodoc 4 -p complex dot dpf. So this is the docking parameter file that we saved. So dpf -l complex dot dlg. So this is the docking log file. So this is the command autodoc 4 space -p complex.dpf space -l complex.dlg. So dlg is docking log file. So it will be running for some time. It will run, and then we can visualize the results tomorrow. So this is how we perform docking using molecule docking. I know it is a series of it has a series of many steps, but there's no other go. It's a very good software, and it is all the output that we get is in a publication format. It is accepted by any journal. So, and it is not commercially available. It is academically free licensed software. So that is why, if it is the commercial software, it might be more user friendly, some other softwares. But here, since Autodoc is a very standard tool and it is free, we need to follow all these steps. Quickly, I will just recap all those things, and then we can go for the question session. So first we need to read the molecule, and then we need to remove water molecule and add hydrogens from edit menu. Then we have to save the file in PDB format. Then we need to open the ligand file, and then we need to save it in PDB-QT format. And then we just go to grid file, we choose the macromolecule, then we choose the ligand molecule, then we set the grid box. Grid box we need to center along the binding site residue. We need to check the binding site coordinates from the PDB file, and then we need to enter the coordinate information in the grid box, and then we need to save the file using .gpf extension. GPF is grid parameter file. This file only we use it for running our autogrid program. And then going to docking menu, choose macromolecule. We have selected the file name of the macromolecule. Then we choose the ligand, and then the search parameters will be genetic algorithm, and docking parameters we will be accepting the default, and then we need to save the output file using Lamarckian genetic algorithm. This is Lamarckian GA using .dpf extension, that is docking parameter file extension, and then we need to run the program. So that's all for today's session. If you have any doubts, we can discuss for a few minutes, and then we can wind up. And all these analysis we can do it tomorrow.
Yes, ma'am. Regarding this, whether we will get any notes or any procedure protocol, something like that?
If you want, we will share tomorrow, sir. No issues.
Please, ma'am, because it was so fast, I am not able to
Yeah.
understand.
Yes, understand.
So
Yeah.
few points.
Yes, sir. Yes, sir.
So if you be providing the protocols, or if you are sharing the slides along like protocols wise, it will be very good, ma'am. Please.
Yeah, sure, sir. Sure. Slides also we have. Slides also we'll share, sir. Maybe the protocol also we will share.
Sure, ma'am. Thank you, ma'am. Thank you.
Thank you, sir.
Ma'am, I have a doubt, ma'am.
Yeah, please.
Ma'am, can you explain the spatial coordination concept, ma'am? I couldn't understand it.
Spatial coordinate information.
Yes, ma'am.
Okay. So we need to orient our grid box along the binding site residue only. Okay. So because if you do a blind docking, what happens means your ligand will bind at any. So some many proteins they will be. Suppose if it is an enzyme, you will have along with active site. You also have an allosteric site also. The software will not be able to identify which exactly is the active site. It knows to identify the cavities. It knows to identify whether the properties of the molecules are opposite complementary to the ligand or not, but it will not exactly know which is the active site. So we need to mention the coordinates of the active site. So for that only what we did was we identify the binding site of that particular target, and then we gave the coordinates of the binding site as the center point for the grid box, so that the ligand will identify the binding site is around that region, and it will try to orient itself to bind to the active site.
Thank you, ma'am.
Is that clear? Yeah.
Yes, ma'am. Thank you.
Yeah. So any other doubts? So
Ma'am,
today we have,
what is the use of torsional potential, ma'am?
what is the use of torsional potential? Yeah. So I told you the ligand will be binding to the receptor by taking different conformations. It cannot, it is actually the ligand will be flexible while doing docking. It will take different orientations depending upon the rotatable bonds only. So in molecules, we have some bonds will be fixed, some bonds will be rotatable bonds. So the flexible molecules, they try to orient in space in different conformations depending upon the torsions only. So that rotatable bonds, what type of angle, at what angle it is rotating, that we call it as the torsions.
Okay, ma'am.
Yeah,
Thank you, thank you, ma'am.
yeah, thank you.
Whether the torsions plays important role in predicting the 3D structure, ma'am?
Torsion does not play important role in predicting 3D structure. It plays an important role in the binding affinity actually. So if the ligand is the orientation of the ligand, that plays a very important role in having the affinity with the macromolecule. So if the orientation is not proper, the affinity will not be good enough.
Okay, okay. Thank you, ma'am.
So all the participants kindly fill up the attendance form for today also. I'm very happy that many of you are active throughout the sessions. I know it is a series of many steps that is involved here, but this is a very good software. If we learn for once, if we try it for once, then it will be useful for you for throughout your career. So that is the ultimate goal. We will be sharing the slides that we have prepared also. In slide itself, I have given step-by-step procedure. If possible, we'll also prepare one study material so that you can have a look at it later on also. So if there are no queries, let us wind up the session for today. Tomorrow, let us all meet at 5:50 p.m. again, and we will check the results of Autodoc, and then we will analyze how the binding has taken place. So thank you all. Thank you very much.
Thank you, ma'am.
So I think my presentation is visible.
Yes, ma'am.
Thank you. So yesterday we performed molecular docking. We had a small molecule, Barangen, and its pharmacological activity was predicted on day one. It was showing antineoplastic activity, and we identified the drug target for that particular Barangen from drug bank, and we have
Excuse me,
generated.
ma'am. Sorry to interrupt. We
Yes,
cannot
sir.
see the screen actually. It is blank. Yes,
Now,
now
sir?
it is coming. Yes, ma'am.
Yeah.
Yes.
Thank you so much, sir. Thank you. So we have docked the both the target and the small molecule, and we have finished our docking process. Today we will be visualizing the docking results, and we will be seeing what interactions have taken place between the target and the small molecule. So, so this part we call it as analysis of the docking results. So here yesterday, I think if we recall what was taught yesterday, finally we had generated one docking parameter file. So after running the command, there will be an output file. So that will be a docking log file with an extension DLG. So today we will be opening that particular file in Autodoc. In Autodoc, we will be opening the DLG file. It is not the DPF file. We will be opening the DLG, that is docking log file, and we will be loading the confirmation, and we will open the macromolecule, and then we will save the confirmation which is having a minimum binding energy. And this confirmation we will be saving it in a format called PDB format, and then we will visualize that particular complex in a software called PyMol. So this is what we are going to do today. And then after visualizing it in PyMol, to know the exact interactions, which are all the residues that is involved, there is another tool called a lig plot. We will be plotting that particular PDB, and we will see which are the amino acids that is involved in the interactions. So before going into analysis, I just wanted to give definitions of few terms that we will be using for um, interpreting our docking results. So Autodoc gives a series of values after the docking process. The first important value that we will be receiving in Autodoc will be a binding energy. So this binding energy is nothing but it is the Gibbs free energy calculation. So it is the sum of the intermolecular forces that is acting between the receptor and the ligand complex. So it generally tells us about the affinity of the ligand and the protein complex. So how much affinity both the small molecule and the macromolecular target is having, depending upon that, we will be having the binding energy. So this is generally calculated based upon Gibbs free energy calculation. So this is one important parameter which we will be using for interpreting our Autodoc results. And the second important parameter that we will be receiving will be ligand efficiency. So ligand efficiency is it tells us about how much binding energy is there per atom, per heavy atom. So excluding the hydrogen, what is the efficiency of each and every atom to bind to the protein? So that is being got by a value called ligand efficiency. So both binding energy as well as uh, ligand efficiency will be in negative terms because it is a free energy value. So these two terminologies will be the result of Autodoc. And the third important parameter that we will be getting from Autodoc will be inhibition constant. So I think most of us might be knowing. So it tells us about how potent an inhibitor is. So it is the concentration that is required to achieve an half maximum inhibition. So here we will be the Autodoc will give us the inhibition constant also. From that also we will be knowing how much dosage of this particular small molecule is required to achieve half maximum inhibition of the particular target that we have chosen. So this is also an important parameter. So the first important parameter that we have to take into account to interpret our docking result will be binding energy. The minimum binding energy will be the least binding, the complex with the least binding energy will be the most stable complex, because we all know, so the free energy value should be very less to get a more complex, more stable complex, as well as the minimum binding energy tells us how much affinity the receptor and the ligand is having with each other. And the second important parameter that we'll be using is inhibition constant, that gives us the concentration that is required to achieve an half maximum inhibition of the target that we have chosen. Next, we will be having some extra energy terms like intermolecular energy, that is the energy that is existing between the molecules. Desolvation energy, this is the electrostatic and van der Waals energy between the ligand and the protein. And along with the solvent, if after binding, what happens? Next, we also have an electrostatic energy, that is the non-bounded energy, and it is due to charges. And then we also get an energy terminology called total energy, which will be the sum of all the energy terms that is happening between both the molecules, and torsions that will be related to the dihedral angles or torsional angles that is existing between the macromolecule and the small molecule. And one more important thing that will be that also will be given in the autodoc, that is the hydrogen bonds. So it also represents how many hydrogen bonds have actually occurred between the macromolecular target and the small molecule. So excluding the least energy values, three important parameters help us in deciding the binding between the macromolecule and the small molecule. One is binding energy, next one is the inhibition constant, and the third one is number of hydrogen bonds. So depending upon these three only, we will be deciding which conformation to take. And finally, we will be visualizing the complex in a software called PyMol. It is actually visualization software. We can visualize the docking results in Autodoc itself, but Autodoc does not have the option for presenting the results in a clear format. So we cannot zoom in the direct ligand separately and where the binding has exactly taken place, what is the surface representation, all those things we cannot do in Autodoc. It is so if you want to do for publication, you will be requiring good quality pictures, which can be generated only by PyMol. So we will be visualizing the complex, the bound complex using PyMol software. So this finally, to get the exact residues that is involved in the involved in the interaction between the target and the small molecule, we will be using a software called LigPlot. So this software is also free, but one thing is we should have a domain email ID. So it is completely free for the academic people, but you should have an academic email ID. Only then it will allow us to download and install the software. So with that, so for all the other softwares, academic email is not mandatory, but for LigPlot, academic email ID, academic domain is mandatory. So only then they will send the license for this particular software. So what is the use of LigPlot is it shows us the exact residues that is involved in the interactions, which are all the atoms. It is exactly. So actually, Autodoc tells us which are the where the hydrogen bond is taking place. So apart from that, we can also see where hydrophobic contacts are there. So we can exactly pinpoint which residues actually interacting with each other, and it will give us a very good scheme. So that scheme diagram will be useful for us to actually present the interactions in a better way. So that's what we use LigPlot. So we'll go on to the demonstrations. Let me open the Autodoc. Okay. So I think yesterday we were running the commands, and I will show that particular working directory. I think all of you remember that all our output files have been saved in this particular folder. So after running the autogrid and autodoc commands, these many files will be automatically generated. So so this one file, this complex.dlg. So this is the output we got after doing the docking, that is docking log file. This file only we are going to open in autodoc, and then we are going to analyze our results. Okay. So So after issuing the commands, kindly open the working directory and see whether all the files have been generated. So if these files are not generated, it means that our command has not been properly executed. There is some error that has occurred, and then we need to rerun all the commands, and then only we can start doing our analysis. So now so let us open. So how to open the analysis? So here yesterday we were we were using this file menu to load our macromolecule, and we were also using this ligand menu to upload our small molecule. We were using the grid menu and docking menu to generate the grid files and docking parameter files. Now we are going to use the analyze menu because we are going to analyze the docking results. We will not be using any of the menus in the top row. We will be using this particular analyze menu only. So just click analyze, and then in dockings, so as we will be using uh, open. So we are going to open the DLG file. I'll repeat once again. DLG is the docking log file that was generated as a result of running the autodock command. So that file we will be opening. So where is that file? So that is in desktop. I have saved in docking folder, ligand folder, so complex.dlg. So once you click open, the DLG file alone will be available. So just click complex.dlg and then open. So this is the small molecule. This is the bar engine. I think from PubChem we downloaded this. This is our bar engine. So in this particular orientation only docking has taken place. So that is what is there in the complex.dlg file. Now we need to load our macromolecule. So first, after loading our docking log file, we need to load our macromolecule or our receptor protein. So for that, what we have to do is just click analyze. From macromolecule, we need to click open. So analyze macromolecule open. So it will give us a warning that it is not found in the directory. So we need to manually select it. So just click OK. So where is the file? In desktop, docking, ligand folder. So receptor. So this is the receptor PDBQT file. This we need to upload now. So receptor.pdbqt is open. So now we have got our macromolecule. We have got our small molecule also. So if you see, there are many options here to do that. So next, what we are going to do is we are going to load the docked conformation. So now analyze. So first we have from analyze menu, we have opened the dockings, open, we have opened the DLG file, then analyze macromolecule, open, we have opened the receptor file. Next, what we are going to do is we are going to generate conformations. So click conformations, load. Okay. So you will get a conformations list like this. So Autodock, it presents 10 different conformations, 10 different docked conformations for this particular complex. I told you yesterday, so the ligand molecule will try to orient itself in different shapes. It will take different orientations, different conformations, and in which particular conformation it is able to bind to the binding region. And after binding, which is having a stability, that particular conformation, the top 10 conformations, which is producing a more stable complex, that will be given as the result in the Autodock. So Autodock generally produces 10 different conformations, 10 different docked conformations. So why we are getting 10 different docked conformations means the ligand molecule will take different conformations and it will try to bind to the binding site of the protein. An Autodock will rank all the 10 conformations, all the conformations, and the top 10 conformations will be given as the result. So now we have loaded the conformations. Now let us play with this particular conformation. So just go to conformations, click play ranked by energy. So this will automatically sort the conformations based on the binding energy. So binding energy, based on the binding energy, it will rank and then it will be loading the conformations. So let us click play ranked by energy. So you will get a play menu like this after this. So till now what I have done, I will repeat once again. Just first go to analyze. From dockings, we have clicked open to open the DLG file. What is the second step? Second step will be analyze. macromolecule open. So we have opened the receptor.pdbqt file. We have opened the macromolecule. What is the third step? Third step is go to confirmation, analyze, confirmations, load. Once you load, all the 10 confirmations will be loaded. Next, we are going to play with the confirmation. So confirmations, play, ranked by energy. So once we click play ranked by energy, we will get a play menu like this. We have the play button, next button, previous button. Like that, we will be getting many options here. So before using the play button, let me from the ligand, let me color the ligand. I hope all of you can see the ligand inside this particular protein. It's colored. Okay. So now please have a view on this particular site. This is the binding region and there is our ligand that is bound to that. Now once I click the next button, the confirmation of the ligand will change. It was not visible, I think. Third confirmation, fourth confirmation, fifth, sixth, seventh, eighth. There's a very small variation, so it is not actually visible. I can see here, but I don't know whether it is really seen in your screens or not. I will play once again. Now I'll go previous. So this is the 10th confirmation. So 9th confirmation. Small change in the position will be taking place. I will zoom a little bit also. I think now you can see. I think now it is visible. You can see small change in the confirmations. So it will change the confirmation and it will bind to the binding site. So 10 different confirmations are generated. So this way we can see how it is bound. But we have to find out which one confirmation, what is the energy, what confirmation value, what are the hydrogen bonds it is formed. All those informations if we require, there is an option button here in the play menu. So we need to open that particular option. So click on that. So this is the option button here. So it will give you show info. Please click show info. And then also click build hydrogen bonds. Okay. So we will get many windows like this. Yeah. So first click build hydrogen bonds and then click show info. Otherwise, hydrogen bond is not displayed here. So now this is confirmation 1. I think you can all see here. This is confirmation 1. So these are all the different parameters it is doing. So binding energy is minus 4.56. Ligand efficiency minus 0.19. Inhibitory constant is 452.91 and the constant unit is mu m, that is micromolar. So 452.91 is the inhibitory constant for this particular barangin. The intermolecular energy, van der Waals desolvation energy, electrostatic energy, total internal energy, torsional energy, and unbound energy is also reported. And finally, if you see the complex, it is forming one hydrogen bond. So the ligand, the oxygen that is present in the ligand molecule, it is forming an hydrogen bond with the gamma hydrogen of serine 478. So it is here in our receptor. There is a amino acid that is serine that is present in 478 position. The gamma Hg is nothing but gamma hydrogen. In the gamma hydrogen is forming a hydrogen bond with the oxygen present in the ligand molecule. So this information is got in Autodoc. So here binding energy is minus 4.56. Now let me move on to the next confirmation. So confirmation 2, you see. So this is minus 4.54. So previously we were having minus 4.56. Now we are having minus 4.54. So we will be for second confirmation also we will be having all the binding energy, all these parameters, and we also will have the hydrogen bonds. And if you see the third confirmation, so here also we are having the value, but the binding energy value will be increasing. So since we have play ranked by energy, the first confirmation will always be the one which is having the minimum binding energy that will be reported. So all the 10 confirmations, we are having a value between -4 somewhere and -4 only. The main important question here is which is the value, or what is there any threshold value for fixing the affinity between the macromolecule and the small molecule. So here docking process is generally a relative process. We don't say that -4 is a very good binding and -10 is very less binding. Affinity is very less. We don't do that way. We do it in a relative comparison way. So how we do it in relatively we compare is, we for all kinds of docking we will be having a standard. So yesterday also we have downloaded the standard drug for this particular drug target. We need to do a docking with the standard drug, and if we are getting a binding energy really lower than the standard molecule, then we can say that it is having a better affinity with the target than the existing drug. So that is how we compare the results. We don't have any threshold value for binding energy or inhibitory constant or number of hydrogen bonds. Actually, we decide whether there is affinity or not between the target and the ligand based on these three parameters only. How less is the binding energy, how less is the inhibitory constant, and how many hydrogen bonds are formed. Only these three parameters help us to decide whether there is affinity between the target and the small molecule or not. But we don't say that the affinity is very strong when the value is below this or above this and there is no threshold value. If you want to do if you want to know whether the affinity is really high, then you need to compare it with another standard compound, and then you need to check whether it is really better, it is having a better affinity than the existing known compound. So that is how we do the interpretation. So we should not simply say that I have got a binding energy -10, I have got a binding energy -20, so I have a very good affinity. It is not really logical. So when you want to say it is having a higher affinity, then you need to compare with the standard existing compound. So this is how we can do some kind of analysis with the Autodoc. But if you see, we cannot present our results in this format, right? So if we want to publish our paper, we cannot simply say that the binding energy is -4.41, the number of hydrogen bonds is 1. So if you present your result like this, it is not at all in a good format. So we require really good visualization software. So for that only we will be opening in PyMol. So PyMol is it is a very good visualization software. We will be visualizing this particular results in PyMol. So for that, how to generate the output file for this. So now out of the 10 confirmations, we know that the first confirmation only is having the minimum binding energy. So we will be finalizing the first confirmation. So let us go to the first confirmation. Okay. Now I've come to the first confirmation. So why did I decide that first confirmation is the best confirmation? Because the binding energy is minimum among all the other 10, and even all the other 10 confirmation are also having only one hydrogen bond. So this particular confirmation is also having only one hydrogen bond. So I'm confirming that out of the 10 confirmation, this is the best confirmation. Now I need to generate the PDB file for this particular confirmation. So how to do that? So there is a small window here. This window we got when we clicked the options button in the play menu. So now in this set play options window, just click build current. So when you press build current, this particular confirmation will be built together. And then what we have to do is we have to save that particular current confirmation. So how to save that current confirmation? To save that, we need to click write current. So write current. So this also I'm going to save it in the working directory. Working again. So let me name it as confirmation.pdb. So that is very important because PyMol sometimes it does not recognize Qt format. So we will be saving it in confirmation.pdb. and save. Now we need to save the complex. So for that, just click write complex, desktop, working folder, again folder. Let me name it as complex.pdb. Click all files. Then see. So I will repeat these steps. So in the set play options window, we need to click build current. After clicking build current, we need to click write current. After clicking write current, we need to save the confirmation in PDB format. After doing that, then click write complex and then save the complex in PDB format. So this is what we have to do in Autodoc for analysis. So I will just quickly go through, quickly tell once again what all we did here. So in analyze, first we, from the docking, we click open the docking log file, and then we loaded the macromolecule, then we loaded the confirmations, and then we played tracked by energy, and then we got a play menu. From the play menu, we clicked an option, and the set play options window was open, and from that we have clicked show info build hydrogen bonds, and after analyzing all the confirmation, we will be saving the best confirmation by clicking build current, write current, and write complex. And most importantly, we need to save the confirmation and the complex in PDB format. So this is what we do with Autodoc. So now we will open our complex.pdb in PyMol and see how the docking has taken place. So let me minimize this. So this is a PyMol. This is a visualization software. Just go to File, Open. So from our working directory, open the complex.pdb file. Yeah. So complex.pdb. Here it is showing. Okay. So this is a PyMol window. So there are many small buttons here available. So A is for action, S is for show, H is for hide, L is for label, and C is for color. So it is very small. I don't know whether you can see it, but there are around 5 buttons here. One is for action, next one is show, next is hide, next is label, and next one is color. So from the show option, so this doesn't look like a protein because it is displaying in cartoon model. Let me display it in wireframe model. So from, if you click yes, all the menus that is available in show will be available. So now from show, I will click wire. Okay. Now we can see the wireframe model. Now let me hide the cartoon. Yeah. So if I add the cartoon, now we can see the protein separately, and we can see the ligand. It is shown in it's actually stick model. Now we will be zooming it in, bringing in our ligand, and we will visualize. For that, there is a very easy way in PyMol. So in action button, there is an option called preset. Okay. So from the action button, there is an option called preset ligands. So preset ligands. So if we click preset ligands, automatically it will zoom in and show our small molecule. And this dotted blue color line is the hydrogen bond. Yellow color line, I'm sorry. So there is also some other interaction, like I think it is hydrophobic. So we can visualize, we can zoom it and see, and click. And you can also label the amino acids, which are all the amino acids that is involved. So for that you have the L option, label residues. If you click, you can see the residues. So we saw serine 478 was forming the hydrogen bond. So the serine 478 is available here. So it is showing the hydrogen bond. Suppose if you want to take a picture and you want to present it in for your publication purpose, then what you can do is just go to file. So there's an option called export image. So we want it in PNG format. So export image as PNG. So now let me save it as picture 1. Save as picture 1. So it's very simple. So just you have to click the action button. Action button. From there is an option called preset. From there, preset ligands. There is also one more option available. So we can also see a surface representation where you can see how the small molecule has perfectly fit into the cleft of the active site of the protein. So for that, what you have to do is from action, preset, ligand sites, solid surface. I think it is visible. So this the surface is the protein and how it is fit into that, yeah, into the cleft. It can be visualized this molecule. It's a very small cleft. It is perfectly fit into it. So we can also see. We can zoom in. Sorry, we can rotate and see how it has beautifully placed into the cleft of the protein. So if you want, you can take a picture of this also. So this also we can save as a picture. So file, export image as PNG. Let us name it as picture 2. So how to display this one? So from action, preset, ligand sites, solid surface. So previously what did we do? For that, action, preset, ligands. So these are the two options. Suppose in some publications they don't accept black color background, they want it in white. So how to do that? There is a command option here, command line option. So you can type on the command bg_color white. bg_color white. So if you see, the background color will become white. So now if you see the preset ligands, so we will get a very depth queuing effect will be there. The actual 3D effect we will get if we get it in white. So after getting white also we can save the picture separately. So that option is also there. So Pymol is having many different options. We can also generate movie. So that will be used when we do the simulation studies, when you are doing a simulation with this protein, all those things, then we can use the movie options, build options, all those things can be done here. But for docking, we will be using only these two options where we will preset the ligands and we will be presetting the ligand sites to get a surface representation. So here bg_color, whatever color. This is the original screen. So this is how we visualize and we take a picture of the images and then we can present it for our publications when we are doing with the document. Now here also some drawback is there. We don't know exactly the exact interactions. We don't have a schematic representation which residues are actually involving, how the bonding has been taken place. So that we don't know because there is no numbering on the ligand molecule. So these red color, they represent oxygen atoms, but still we don't know which oxygen is actually has bound or has formed the bond in the drug target. So in that case, we need to use the software called LigPlot. So LigPlot, how to get LigPlot? Just go to. So just click Lickplot download. So latest version is Lickplot Plus we have now. So we can have Lickplot Plus download. So here we need to give a email address and sorry, to download the license file. So if you do not have a password, please go to academic license. So first we need to click the academic license and we need to fill the form. Here we need to fill the form. The email address should be an institutional email ID. It should not be Gmail. So once if you have an institutional email ID, immediately they will send you a password. With that email address and password, we can download the Lickplot Plus download file. And we will be requiring Java. Java should be previously installed, only then Lickplot will be opening. So after installing Java, you please install Lickplot Plus and then we will be having a file like this. So after installing Lickplot, you will have a window like this. So here we need to open the PDB file. So which PDB file we need to download? We need to open. So in Autodoc, we have done the docking analysis and we have saved the protein in PDB format. We need to open that particular protein and then we can visualize the. So just go to File, Open, PDB file. So it is asking for a PDB code. I'm sorry. Sorry, there's a browse button here. Just click browse. This Lickplot is not only used to visualize the interactions of the docked complex. Suppose if there is some complex in the PDB itself and you want to visualize that particular interactions also, we can use this Lickplot Plus. So docking, ligand, and here we have the complex. So this is our PDB file that we have saved today now. A few minutes back, we have saved from Autodoc. That we open. Now run. Okay, see how beautifully the picture has come. So this is violet color one. It is the so this violet color one is the ligand that we are having. All these residues, these are all the residues which is having having interactions with this particular ligand molecule. If you see here, these arc that is pointing towards the ligand, they represents the hydrophobic interactions. So these are all hydrophobic interactions. And here, this arc that is shown around this atom, that is also hydrophobic interaction. So these atoms, and for example, arginine 192 of the A chain, it is having hydrophobic interactions with this particular ligand. And the hydrogen bond, if any, that will be represented as dotted lines. So this hydrogen bond is not. See. There is some problem here. So actually there is a hydrogen bond. Serine 478 is not. Serine 478 is here. So that this is forming a hydrogen bond with this oxygen. This will be actually shown as a dotted line. So this arc they represents the hydrophobic interactions. The hydrogen bond will be denoted as dotted lines. I'm sorry, it is not. I don't know why it is not coming today. because serine 478, we know that it was having a hydrogen bond with this oxygen, I guess. So it should be a dotted line that is coming from this particular oxygen to this particular serine 478. So that dotted line that represents hydrogen. So this Lick plot, it helps us to exactly pinpoint which are all the amino acids that is interacting with the ligand molecule, and the two important interactions, that is hydrogen bonding and hydrophobic interactions alone, will be displayed, because only these two interactions that happens between the target and the drug, because all the strong interactions does not take place with the because the binding should be short. It should be for a short span of time only after, because after that it has to be excreted, and it has to come out of the body, otherwise it will become toxic. So these very weak interactions such as hydrogen bonds and hydrophobic interactions alone will be taken into account for while doing the docking. So hydrogen bond will be displayed as dotted lines, and hydrophobic interactions will be shown as outward arcs. So this is how we can visualize. Suppose if we want to save this result, so what we can do is we can go to file, and there is an option called write PS file. So this is a Lick plot picture. So Lick plot, and we can save it. Okay. And then you can use it for your publication. This picture we can directly take it and we can use it for our publication. So this is how we present our results. So that's what that's all about analyzing the docking results. I'll just have a quick recap, and then we will have the question session if you have any clarifications. So after running the Autodoc commands, we have to open the Autodoc tools, and then we have to visualize the docking confirmations by loading the docking log file, and we have to load the macromolecule. Then we have to load the confirmations, and we have to play rank by energy. Then with the help of the play options, we need to build the hydrogen bonds, and we have to show the information, and all the binding parameters, binding energy parameters, all the parameters, docking parameters will be displayed. And then we have to save the best confirmation and the best complex. So we have to build current, and we have to save the current confirmation, and then we have to save the complex also. After saving the complex, we need to open in PyMod, and then we can visualize the interactions. We can display the pictures of interactions that is taking place, and same PDB file we can load it in Lickplot to exactly visualize the interaction, which are all the actual residues that is involved in both hydrogen bonding and hydrophobic interactions. So that's all about molecular docking. If there are any clarifications, we can have a question for a few minutes.
Excuse me, ma'am.
Yes, sir.
Yesterday, you have just run this particular thing in the command prompt.
Yes, sir. Yes, sir.
So how much time we have to keep? That is, it will be running for throughout the night?
No, sir. No, sir. Yesterday, once before I closed itself, it ran, sir. It got over.
Okay.
Yeah. It
Usually, if we are having multiple samples for docking,
yes,
so how much time it will take, ma'am, if you are running with command prompt?
Multiple samples also, sir. Actually, it takes less than 5 minutes only, sir, to run. How much if you're doing blind docking, suppose you don't know the active site and you're not centering your grid box around the active site, means then for blind docking only it takes minutes of time. If you know the active site and you're giving the grid box, centering the grid box exactly along the binding site, means it takes less than 5 minutes only, sir, for one ligand molecule.
Okay, so immediately after the command prompt, we have to open the Autodoc and we can perform this. Whatever.
Yes, sir. Yes, sir. Yes, sir. Immediately after the command prompt, you can open it and visualize it, sir. Yesterday, it was already a series of commands, so I did not want to confuse everyone with the same. Immediately after running the command, once the command has been executed, we can open the Autodoc and we can do whatever I have done today. It can be done immediately. You need not wait. Yes.
Okay, ma'am. Thank you, ma'am. Thank you.
Thank you, sir. So, dear participants,
Excuse me, ma'am.
yes, ma'am, yes, ma'am.
Ma'am, I'm having a doubt. I just need a clarification. One thing that if I did in GC and I am getting nearly 100 compounds and I am docking all the 100 compounds, the top scoring compound is not showing the hydrogen bond interaction means whether I could consider that or not.
Ma'am, hydrogen bond alone is not a criteria for deciding the docking, no, ma'am. So,
Okay, ma'am.
if your compound is having a better binding energy than the standard that you are referring to, then that compound can be taken into account, ma'am. Hydrogen bond alone is not a criteria for selecting or filtering any compound for docking, ma'am.
No, ma'am, I didn't docking. Actually, I gone for a training, ma'am. So there, what we did is, so we have taken all the PDB
All
files.
the
Yes, ma'am. In a docking, after completing a docking, we'll get a dock score, no, ma'am.
yes, ma'am, yes, ma'am.
So that dock score la வந்துட்டு top score compound I gone with actually I researched in picture and I took a picture for knowing the interactions. There what I have understood is there is no interaction at all. That is one category, and another one category is there is no hydrogen bond interaction. So what they taught us is hydrogen bond interaction must be there. Then only we can consider the docking that compound is uh, eligible for going with further pharmacological actions like they told. So is it correct or not that I need
Okay,
a clarification? I need your
okay.
opinion in this regard, ma'am.
Okay, ma'am. Okay, ma'am. When you are comparing compounds, it is all. Suppose if you get some other compound with hydrogen bond, it is always better to go with the compound which is having hydrogen bonds only. Because without hydrogen bond, if you're going for the next step of pharmacological, doing it, taking it to the pharmaceutical company and for synthesis or and all means, then it will not be that good. So it is all. If you don't get a hydrogen bond, even if 10 conformations we got today, no, ma'am. So today in all the 10 conformations we got one hydrogen bond. If there were more than one hydrogen bond in any of the other conformations, means I would have chosen that particular conformation only as the best conformation. So more the hydrogen bonds or the presence of hydrogen bond will also be an important criteria, but that alone will not be an important criteria. That's what I want to tell you.
On certain compounds, they didn't show any of the interaction. So, for example,
If it is
the
not
compound
showing any interaction means then it will not bind, ma'am, actually.
that's what, ma'am. It's not at all binded with an ligand protein interaction. It's not happened there. So we may not be take that, no, ma'am.
Yes, ma'am. Yes, we should not take that. Yes, ma'am. For the next step of analysis, we cannot take. Yes, ma'am. You're right.
Okay, ma'am. Okay. Ma'am, another one thing is, if we are going for modeling means, so that iTester and this one, Pyri 2, we are using an online tool for modeling the proteins, no, ma'am?
Okay, ma'am. Yes, ma'am. I tested.
iTester. So in that, what while I'm trying with an iTester, what happened is it is accepting a particular length. So more than that, it is not accepting the length of the protein for modeling, ma'am. So
What is the length of your protein, ma'am?
that அந்த அளவுக்கு I didn't notice it, ma'am. But while I am giving it, so what it told is your length is too high. We cannot able to do it like it told. So my question in this regard is, compared with Pyri, if we are going with an iTester for modeling and protein means it will give a better modeled protein like I studied in papers. So is it so, ma'am?
Ma'am, actually, I have not personally worked with Pyri, ma'am, but I have worked with iTester. But this is the first time I'm getting a query like this. That is, it is not accepting a very big length protein. iTester, I have really modeled around hundreds and hundreds of different proteins. None of the protein which showed that kind of error,
Okay,
ma'am.
ma'am. So for this, I'm not sure within size. Whereas the protein what I have tried is chitin synthase and glycan synthase. These are all the two proteins I have tried it in an iTester. So while I am pasting in sequences, it showed that your sequences is beyond the length. So we may not be able to model using that like it said.
Okay.
And I heard the iTester is giving a better model structure than Pyri like. So
Okay.
I just need a confirmation in this. Is it so, ma'am? iTester is a better one for modeling compared with Pyri, ma'am?
Yes, ma'am. iTester, it is the best to online software for doing molecular modeling, ma'am. Even very big research institutions, they are using iTester only, ma'am, because it is user friendly also. So we also have many offline softwares But that and all requires so much of computational knowledge, ma'am. So this iTester is really a very user-friendly software. It is online. It is the best one as far as I know, ma'am, till now, which is online, ma'am. So if it is giving some error, in that case, maybe we can use some other software, but it is the best one, ma'am. There is no doubt in that, ma'am.
Thank you, ma'am. Ma'am, one more question.
Yes, ma'am.
Ma'am, while in that training, what I did is, so I have downloaded a structure from PDB and I saved it as SDB file format something.
STF.
Yes, I'm not sure about that, ma'am.
Yes,
So
okay.
I'm basically from a biotechnology. So just now I started learning the bioinformatics.
Okay, ma'am, no
So
problem.
ma'am, then what I did is, so the same file what I have saved is actually the workshop they did it in Glide, and they have taken all the things as it is as a STB file for ligand preparations.
Okay, okay.
Ligpre, that file will come now, ma'am. So for that they have taken the file as it is. So while you are taking your in your session, what you told is that the STB format has to be converted into PDB. Then only we can able to take it up the things for docking, like you said.
Yes,
So and could you please clarify in this, ma'am?
sure, ma'am. So that is the difference between the commercial software and academically free licensed software, ma'am. Autodoc is a free software. We need not pay Glide and all. We need to pay and we have to purchase, ma'am. So since it is a commercial software, it is more user friendly. We need not convert into one file format into another file format. We can easily use any file format and we can perform our docking. So, but since these are all so for performing simple docking, or so being a life science people, we will not continue all the time. We will not be doing docking. So purchasing a software for lakhs and lakhs of rupees and keeping it in our lab is not going to help us. So in that case only we are using academically licensed software, which is of publishable quality. So these softwares, they don't have formats. It does not accept any format. It requires only specific formats, ma'am. That is why we are converting from SDF into PDB. That's the difference, ma'am. If it is a commercial software, it supports any file format.
Understood, ma'am. So according to the software, we are changing the file format.
Yes, ma'am. Yes, ma'am. Yeah.
Okay, ma'am. Thank you, ma'am. Thanks a lot.
Thank you, sir. Thank you. So any other question?
Ma'am, if you
Yes.
don't mind, could you please explain me the inhibition constant part, ma'am? Like, what is the role of inhibition constant in this binding, ma'am?
Inhibition constant is the dosage of drug that is required to give. So if you want to give, if you want to get 50% inhibition, then how much dose of or how much concentration of the small molecule we need to use? That is what is got from inhibition constant.
Okay, ma'am. Thank you.
Yeah. It tells about the drug concentration. How much concentration of the drug is required to get a 50% inhibition with the target.
Yeah, ma'am. Understood. Thank you. Thank you.
Yes. Yeah. Participants, since some of you have requested, we have also prepared some workshop materials, and we have also uploaded in our drive, and that drive link is also shared with you. Unfortunately, we could not record the first day session. There was some technical issue with our domain, and we could not record the first day session. It's like yesterday's session, it has been recorded and it is also uploaded in the drive, and the link is also shared with you. And all the 3 days PPT is also shared, and yesterday's protocol is also shared in your drive. Today's video and today's protocols will be shared before tomorrow morning. So you can have access to all the study materials, whatever protocols that we have discussed for the past 3 days. Because since all these learning should not be left with this 3 days itself. So to your maximum benefit, we have tried our best to simplify the study materials, and we have also uploaded in the drive. Kindly make use of it. And I wholeheartedly thank all the participants. It was actually really an overwhelming response from the participants, not only from life science background. Many other participants all over the country have shown interest, and they have also actively participated. So on behalf of our college management, principal, and secretary, I wholeheartedly thank all the participants for patiently listening to the lectures for the past 3 days and clarifying your doubts. and I wish you all the best for your career and your future.
Thank you, Sivashankari ma'am. I am Aarti from Krishna Arts and Science College, ma'am.
Yes, ma'am. Thank you, ma'am. Thank you so much. Thank you for your participation, ma'am.
It's a very informative session, ma'am. Ma'am, you just conduct a hands-on training.
Yeah, sure, ma'am. Yeah, sure,
In
ma'am.
the offline mode.
We will organize, ma'am.
That we are.
Definitely, ma'am. Definitely. That will be more better,
Hello,
ma'am.
ma'am.
This is the online.
Hello, ma'am.
Yes, ma'am. I can hear you, ma'am. I can hear you.
Ma'am, can you able to hear me? Hello,
Yes, ma'am.
ma'am.
I can hear you, ma'am. Yes, ma'am. I can hear you, ma'am.
Hello, ma'am. You can able to hear me, ma'am?
Yes, ma'am. I can hear you, ma'am. Please tell me, ma'am.
Ma'am, soon you just have a hands-on offline mode, ma'am, training. That will be somewhat good for our students. More informative, and they can have like a practical session also. Then
Sure,
and
ma'am.
there.
Definitely. We'll arrange one in near
Sure.
future, ma'am. Surely, ma'am. Your suggestion is well taken. It will be arranged, ma'am.
And also, ma'am, they next when you are taking next session, ma'am, you just tell more about the QSR and all, ma'am. That will be more useful for them, no, ma'am, when they are having their chem sketch.
Yes, ma'am. Yeah, sure, ma'am. Sure, ma'am. Definitely. Yeah.
Okay, ma'am. Thank you so much, ma'am. Thank you.
Thank you. So all the participants kindly fill the feedback link also. Your certificates, you will be receiving your certificates by tomorrow evening. Kindly fill your feedback forms, and if there are no queries, we can wind up the session. I call upon Ms. Bhuneshwari for the formal vote of thanks.
Yes, ma'am. A warm welcome and pleasant evening to one and all present here. On behalf of Nirmala College for Women, Department of Bioinformatics, I take this opportunity to propose a vote of thanks. First, I would like to extend a special thanks to Reverend Dr. Sister Ruby Alangara Mary, Secretary, Nirmala College for Women, for her wishes and prayers. I wish to express my gratitude to our principal, Reverend Dr. Sister Helen, for her constant support. And I am very grateful thanks to Dr. Sister Mary Fabiola, Vice Principal, Nirmala College for Women. And I extend a very hearty vote of thanks to our ma'am, Dr.S.Sivashankari, Department of Bioinformatics, Nirmala College for Women. And I also thanks to Reverend Dr. Sister Emelda Mary, Controller of Examination, Mrs. Polentia Kamal, Dean of Academics, Dr. U. Mary, Dean of Students Affairs, and Dr. Pauline Vasandi Joseph, IQAC coordinators. I thank all the participants for active participation. Once again, thank you all.
Thank you, Ma. Thank you very much. Thank you, one and all, once again. Wish you all success. Please fill the feedback form and you can please leave the session. Thank you very much.
Thank you, ma'am. Thank you.
Thank you, sir. Thank you.